Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Central University's Common Entrance Test or CUCET MSc Entrance Exam question paper for the subject Applied Microbiology. This was conducted in the year 10th June 2023. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of Applied Microbiology for the same entrance examination, you can go to my channel Omni Bio Entrance, see for the playlist. In the playlist, select the folder CUCET, you will find the question papers there. Coming to the first question. Which of the following scientists demonstrated that crossing over involves physical exchange between chromatids? So it was Barbara McClintock who demonstrated that crossing over involves physical exchange between chromatids. The symmetry of head in T4 phage is, so it is icosahedral symmetry. Answer is option 1. Which of the following is a eukaryotic microorganism? Bacterium, cyanobacterium, algae, archaea. So all of you know it is algae that is a eukaryotic microorganism. Answer is option 3. Then T4 phage follows dash cycle. So T4 phage, it is an example of lytic cycle. Answer is option 1. UV light causes formation of following dimer in DNA. So what dimer? It causes thymine dimer, TT dimer. Answer is option 3. Methanogens belong to which group? So methanogens, they belong to archaea. Answer is option 2. All these are direct question, that's why I am not explaining much. The generation time of E. coli growing in nutrient broth at 37 degrees Celsius is. So E. coli, it has a generation time of 17 to 20 minutes or 15 to 20 minutes. So in nutrient broth at 37 degrees Celsius, answer is option 2, that is 17 minutes. What is a prophage? Prophage is seen in the lysogenic cycle of a bacteriophage. So in that, the bacteriophage DNA, it will get integrated into the bacterial genome, and that stage is called as the prophage. So answer is option 2, that is bacteriophage DNA integrated into the bacterial genome. Answer is option 2. Beta-galactosidase gene inducer is. So in lac operon, you have got the beta-galactosidase enzyme being produced, and there is a gene for the production of this beta-galactosidase enzyme, and beta-galactosidase, it breaks down lactose, and which is the inducer of beta-galactosidase gene? It is allolactose. Answer is option 4. Then in the transversion mutation, so you can remember it easily. Here the version is changed. In transversion, the version changes. That is, purine gets converted to pyrimidine, or pyrimidine get converted to purine. Whereas in transition, there is no version change. Purine to purine, one purine substituted by another purine, or a pyrimidine substituted by another pyrimidine. That is in case of transition mutation. Here version changes. That is transversion. So here the answer will be purine is substituted for a pyrimidine. Answer is option 1. Then next one. Which of the following cyanobacteria is not used as a biofertilizer? This question was a drop question, but I have answered it. The question should have been, which of the following cyanobacteria is used as a biofertilizer? All are used as biofertilizer. That is Nostoc, Anabina, Stegonema, Cyctonema. All are used as biofertilizer. And one more option should have been there, all of the above. If the question was, which of the following cyanobacteria are used as biofertilizer? Okay, next one. Which one is the non-symbiotic biofertilizer? Here answer is Azotobacter. Azotobacter, it fixes atmospheric nitrogen into soil without forming symbiotic relationship between or with plants. Then Anabina, it forms association with Azolla, a water fern. Azolla, it is a symbiotic host for Anabina. Rhizobium, it forms root nodules or in leguminous plants. And it is involved in symbiotic nitrogen fixation. Then next one. In microbial interaction, if one organism adversely affects another organism, it is known as first option, commensalism. What is commensalism? When one organism benefits, the other organism is not affected. So in commensalism, one organism benefits whereas the other is not affected. Mutualism, both organisms are benefited. Competition, it is an interaction between organisms for shared resources or limited resources. That is competition. They compete for the same resources, that is shared resources or for limited resources. That is competition. So here is the question is the interaction in which one organism adversely affects another organism. It is known as amensalism. Answer is option 3. Then next one. Which organelle is absent in plant cells compared to animal cell? It is centrosomes. Centrosomes are absent in plant cells but are present in animal cells. Then next one. Crossing over take place during which phase of meiosis? So it is at the Packettine stage that crossing over take place. What happens at the diplotin? Here the homologous chromosome begin to separate and chiasmata are also visible. What happens during diakinesis? Chromosomes further condense and separate. Nuclear envelope breakdown. Then metaphase 1, homologous chromosomes align at the equator. Sister chromatids, they remain attached to the centromere and the chromosomes are maximally condensed at this stage. Then spindle fibers also start to arise and they attach to the centromeres. That is what is happening in metaphase 1. Here the question is crossing over taking place. So it is at the Packettine stage of meiosis 1. Then next question. Dominant plant genotype can be determined by. So it is by test cross that you can determine. Suppose this capital T shown in this example is the unknown genotype and small t is the recessive parent. Okay. Now before that test cross, it is the crossing over between an unknown genotype. Unknown genotype is heterozygous or homozygous dominant. And this unknown genotype, it is crossed with the recessive parent. In all the cases, it is the recessive parent it is crossed with. That is, it can be homozygous recessive. So here capital T, that is homozygous dominant, that is the unknown genotype. We don't know the genotype of this plant. It is crossed with homozygous recessive parent. You will get capital T, small t. Similarly, so whenever capital T occurs, the offspring will be capital T, small t, heterozygous. And you will get all dominant character plants. Now if your unknown genotype is heterozygous, that is capital T, small t, it is crossed with homozygous recessive, then you will get a dominant heterozygous condition and a recessive plant. So dominant plant genotype, it can be determined by test cross. Answer is option 3. Now back cross, it is used to transfer a desirable trait from one parent to another. That is for that reason only you usually use back cross. Hybrid cross, it is used to combine desirable traits from two parents. And dihybrid cross, everybody knows, it is used to study inheritance of two genes. Then next one. In one of the Mendel's crosses, he observed 315 round yellow, 108 round green, 101 wrinkled yellow and 32 wrinkled green F2 plants. Identify the type of the cross. So here the least one is 32. So 315 divided by 32 you will get around 9. 108 divided by 32 you will get around 3. 101 divided by 32 you will get again around 3. 32 divided by 32 will be 1. So your ratio is 9:3:3:1. So that is a dihybrid cross. So the answer here is option 1. Cell cycle is regulated by cycline phosphorylation. Cyclines are not phosphorylated nor are cyclines dephosphorylated. Okay, it is the cycline dependent kinases enzymes, cycline dependent kinases that are phosphorylated, dephosphorylated and undergo either activation or inhibition or activation. Anyway, cycline dephosphorylation, that's also not correct. Cyclines are not dephosphorylated. Cycline dependent cycline dephosphorylation, such thing doesn't exist. Protein phosphorylation and degradation, yes, that is the answer. So cell cycle regulation, it involves cycline dependent kinases, cyclines that are binding proteins of the cycline dependent kinases, protein phosphorylation, that is for activation or inhibition of the CDKs, protein degradation and removing cyclines and inhibitors. So here the answer is protein phosphorylation and degradation. Answer is option 4. The disaccharide which gives two same monosaccharides on hydrolysis, two same. Sucrose on hydrolysis, it will give glucose and fructose. So they are both not the same. Lactose will give glucose plus galactose. Maltose will give two glucose molecules. So maltose is giving two same monosaccharides. So answer here is option 3. Lactulose it will give fructose and galactose. Then next one. In which of the following animals environmental factors determine the sex? So it is in the case of turtles. So it shows temperature dependent sex determination. Turtles. So the answer here is option 3. In recessive mutation, gene expressed only when heterozygous? No. Dominant gene will be expressed there. Gene expressed when homozygous? Yes. In a recessive mutation, the gene is expressed in the homozygous condition. The next one. The Taq DNA polymerase from Thermus aquaticus belong to which class of DNA polymerase? So it belongs to class 1. Answer is option 1. In humans, the most abundant protein is naturally collagen. Answer is option 3. The succession process in which microorganisms modify the habitat in a way that permits new population to develop is called. This is usually seen in case of biofilm formation. Okay, so we will see allogenic succession. So community, it responds to external changes. Autogenic succession is community, no external factors triggers the succession. Community succession means changes in the species composition after a disturbance. Secondary succession means changes in the established microbial community. That is, upon intake of antibiotic, there will be change in the gut microbiome. That is secondary succession. Here biofilm formation and all, it will come under autogenic succession. So it is a autogenic succession. It is the process by which microorganisms modify the habitat. Here no external factors trigger. Only internal factors are triggering the modification of the habitat. so that will permit new population to develop. That is called as autogenic succession. Answer is option 4. In vertebrates, fat is stored in the form of triacylglycerols. Answer is option 2. Then next one. Which of the following immunoglobulin class is found in maximum concentration in the blood? Okay, it is IgG, immunoglobulin G. Answer is option 2. In the plasma membrane, the most abundant lipids are, of course, phospholipids. Answer is option 3. In the red light region, which pigment has higher absorbance? Naturally, it is chlorophyll A. Answer is option 2, chlorophyll A. The most abundant enzyme on this planet. Plants are more in number, so naturally it will be rubisco that is more abundant. So answer is option 3. Seed dormancy is due to the accumulation of abscisic acid. Answer is option 3. Bacteria protect themselves by fragmenting viral DNA upon entry by. So they will use their restriction endonucleases which will cut the DNA at specific sequences and resulting in fragmentation of the viral DNA, host modification and restriction. So bacteria, it is by the enzyme endonuclease. Answer is option 1. In reverse phase chromatography, mobile phase is polar, stationary phase, that itself is the answer. Answer is option 1. Mobile phase is polar, stationary phase is non-polar. And here the separation, it is based on hydrophobic nature. So non-polar compounds, they retain longer, that is they will bind to the stationary phase and they will be retained there in the column. Whereas in the polar compounds, they elute faster. So answer here is option 1. Then next one. At isoelectric point, the proteins have no net charge at the isoelectric point. Answer is option 4. In xylem, which type of transport is present? It is unidirectional transport in xylem. That is, there is only one way movement of water and minerals. That is from the root to the leaves through the xylem. There is no downward movement. That is from leaves to root it doesn't go. Only in the upward movement. That is from root to the leaves it goes. So it is unidirectional in case of xylem. Then next one. The term ecosystem was coined by. So it was coined by A.G. Tansley. Answer is option 1. In ecology terminology, the natural residence of every organism is known as. So natural residence is known as habitat. Answer is option 4. Xerophiles. Xerophiles grow in dry place. Answer itself is option 1. The final electron acceptor in lactic acid fermentation is, so it is pyruvate. That is the final electron acceptor. It accepts electrons from NADH and get reduced to lactate. That is pyruvate plus NADH plus H plus gives lactate plus NAD. Then next one. The activity of a species determines community structure in spite of their low abundance or biomass. That species is called, it is called as keystone species. Answer is option 1. The species that first colonize in new habitats created by disturbance is known as, so they are known as pioneer species. Answer is option 3. They are the species that will first colonize a new habitat after a disturbance. Lichens are indicator of, so they are indicator of air pollution, usually sulfur dioxide pollution. Then polygyny means one male mates with several females. Answer is option 1. Humans are K selection and R selection. Okay. Humans are large in body size. So large brain to body mass ratio. Then low reproductive rate when compared to other mammals. They have a long lifespan and there is high investment on the offsprings. That is education, healthcare, all these for offspring. So there is high investment in the offsprings. Then complex social structures and cultures. So because of these reasons, humans are case selection species. So answer is option 1.